Post-mortemly, previously they used to depend upon the presence to diagnose rabies virus by the presence of Negri bodies present in the brain smears of the post-mortemly. They used to take smears from the brain, spread it on slides, stain it by special stain, and examined by the normal ordinary light microscope. By the light microscope, they are looking for the presence of Negri bodies. What are the Negri bodies? Negri bodies are intracytoplasmic inclusion bodies present within the neurons, the spinal cord or the brain, and their presence denotes the presence of the virus or diagnose the presence of the virus. However, in 20% of the cases, the virus may be present, but no Negri bodies are present on the slide. So Negri bodies cannot be observed in 20% of the cases, and therefore their absence does not rule out rabies infection. And we have to look for another method to diagnose the presence of rabies virus. This figure, we have two figures here. This figure represents Negri bodies. This is the brain cells after we have made a take biopsy from the brain, of course post-mortemly after the death of the human or the animal. We take smear from the brain, we spread it on a slide, fix it by special techniques, stain it, and then we examine it by the light microscope. Under the microscope, we see the following. We notice that these are the brain neurons. This is the nucleus of the brain neurons, and then Negri bodies are these small spots surrounding or capping the nucleus. We call it capping. Capping, why? Because it is present always over the nucleus. Capping, زي cap, like a cap over the nucleus. Capping the nucleus. These Negri bodies are intracytoplasmic inclusion bodies. What do you mean intracytoplasmic inclusion bodies? These have been found to be viral aggregates during the replication of the virus. The virus replicates within the cell. We all know that. Some viral particles do not form complete virus particles, but are present in the brain stem, incompletely present in the brain stem. These form circles or bodies called bodies found capping the nucleus of the brain cells. The arrow here demonstrate the presence of the Negri bodies. This is another figure that shows the presence of Negri bodies using different stains. You can see also these are the brain cells, these are the nucleus of the brain neurons, and these dark purple spots represent the Negri bodies or are Negri bodies. Some of them are present intracellular within the brain neurons, but if the cells are destroyed, they are liberated in the surrounding, and you can notice the presence of Negri bodies. Alternatively, the gold standard of diagnostic technique post-mortemly is to detect rabies virus antigen by means of fluorescent antibody test. As I've said, presence of Negri bodies is not conclusive because 20% of the cases fail to demonstrate the presence of Negri virus, or they are not present. They are not present. Why? Because the number of virus particles is too small, so they cannot form the Negri bodies. So a better method to diagnose is the presence of or detection of virus antigen by fluorescent antibody technique in the brain tissue or in the spinal cord or in the cornea or whatever specimen is present in the animal. The direct immunofluorescent technique is a method where you can detect the antigen by attaching it to a specific rabies immunoglobulin, and this rabies immunoglobulin is labeled by a fluorescent dye. If the antigen is present in the specimen, a specific antigen antibody reaction occurs, and this a specific antigen antibody reaction occurs which fix the complex, the immune complex, to the slide of the microscope, and then we can examine it by the ultraviolet microscope. This figure here, we can see a column on the right and a column to the left. The column to the left represents the specimen, the brain specimen, containing or including the virus, rabies virus antigen, or the rabies. We want to see whether it is present or not in the brain smears. The brain smears are placed on slides, fixed by a special technique, and then we add immunoglobulins, specific immunoglobulins, specific antibodies to rabies, which is conjugated by a fluorescent dye. If the antigen is present in the slide, in other words, in the sample, in other words, the brain is infected or the animal is infected, a specific antigen-antibody reaction occurs, which fixes it to the slide. And then by special methods, we wash and prepare the slide and examine it by the ultraviolet microscope, and you see this picture in front of you. You will see fluorescent or luminous object under the ultraviolet microscope, or examination using the ultraviolet microscope. This column, right column here, where the virus has been brain smears has been prepared, overlay the slides, and then prepared and fixed by special techniques, however not containing the rabies antigen. In other words, this patient or this individual, this animal is not infected. When we add the immunoglobulins, the immunoglobulin will not interact with the brain cells because there should be specific antigen present on the slide. So it will be washed away during the preparation technique, and nothing will be present when examined by the ultraviolet microscope. By examining the ultraviolet microscope, a dark field background will be present with demonstrating no luminous object or no fluorescent object as seen in this figure. However, there are several tests now available for diagnosis of anti-rabies ante-mortem. Ante-mortem meaning preparing specimens from living individuals, and we try to diagnose rabies in order to isolate them or to perform special techniques or try to prevent and extend their lifespan. Specimens. What is the right specimens to be taken for demonstration of rabies virus? It's very important to choose or select the proper specimen, because if you don't get the proper specimen, you will get false negative results. The specimens are saliva, skin biopsies taken from the hair follicles at the nape of the neck, the back of the neck, serum and CSF also may be used. So the specimen applied for diagnosis or recommended for diagnosis of rabies virus is saliva, skin biopsies, serum or CSF. The skin of biopsy is taken from the hair follicles at the nape of the neck. What is the nape of the neck? This figure demonstrates the nape of the neck. The nape of the neck is the back of the neck at the end of the occipital bone, or at the end of the hairline. This is the nape of the neck. This place is fully rich with nerve supply, with nerve endings, and the virus, you can find the virus reaching this in large amount. You see these are the sensory nerves, and the virus, after being replicating in the brain, spreads down through sensory nerves surrounding the skin around the hair follicle. So a very rich supply or very rich method for detection of rabies virus is through taking skin biopsy from the back of the neck at the hair line. Serum, blood specimen, obtaining serum for detection of antibodies, and CSF. However, the time of the biopsy influences the results, meaning that the right time is essential, maximum after 4 days after the disease onset. After the disease starts, we take 4 days and then start to take the specimen. I'd like to clarify here how skin biopsy is taken or obtained from the patient. This is an infected patient. We put him on one side, and then through the nape, at the nape of the neck, we disinfect the area, and then we take a biopsy from the skin surrounding the hair follicles. Here also another figure where we obtain the skin biopsy surrounding the hair follicles from the eyebrows, where the virus spreads through sensory nerves, richly supply the skin surrounding the hair follicles, and this is a very rich area where we can detect the presence of virus in large amounts. Okay, after we have taken the proper specimen, we look for presence of rabies antigen or nucleic acid detection. The skin biopsy specimens can be tested for the presence of viral antigens in the neurons in close proximity to the hair follicles. We take a skin specimen, like we explained before, spread it on a slide, stain it with labeled immunoglobulins, labeled by fluorescent labeled fluorochrome immunoglobulins, wash and prepare the slide and examine for the presence of rabies virus. Here, no virus antigen is present, and therefore examination by the ultraviolet microscope appears as dark field or black field with no fluorescence present. This is the ultraviolet microscope, and this is the picture you see by the ultraviolet microscope. Next, if ultraviolet microscope facility is not present, because you cannot always find ultraviolet microscope, we can stain the skin specimens previously obtained, as I've explained, by a special stain known as immunoperoxidase stain. This specimen is fixed on the slide, incubated with specific rabies immunoglobulin, washed by special technique, and then we add peroxidase enzyme. This peroxidase enzyme, when we add the susceptible substrate to this enzyme on the slide, this stains the slide and can be examined by ordinary light microscope. You will see the following figure here. These are two different stains used. In this figure, we can notice the rabies antigen is present in the form of red spots, and these are the cells or cellular components of the specimen. Here, the rabies antigen is present on the form of red spots or blue spots, depending upon the type of immunoperoxidase and the substrate used. Rabies antigen, rabies virus antigen, could also be detected by fluorescent microscope from specimen taken from peripheral nerves, from salivary glands, from the saliva, from the cornea, or from the skin surrounding the hair follicles, and this is called intravitum diagnosis. Stained also by the same method by direct immunofluorescent technique or fluorescent antibody technique. Then we come to nucleic acid detection. We said we can detect the presence of viral antigen either by staining with fluorescent antibody technique or by detecting the nucleic acid, the genome of the virus particle in the specimen selected, in the proper specimen selected. Skin and saliva samples can be tested for the presence of viral RNA by the test known as reverse transcriptase enzyme. During this PCR technique or reverse transcriptase test, reverse transcriptase PCR test, the rabies antigen which is the genome, which is RNA in nature, is transferred first into DNA by special enzymes. The DNA is put in the specimen is put in a special apparatus known as thermal cycler, where there is an increase in the number or amplification of the number of genome of the virus particle. After the virus has been amplified enough, it can be the presence of the virus genome can be detected by staining by gel electrophoresis, and we notice here the bands that characterize the genome of the virus particle. We have a map, and we know the genome of the virus, rabies virus, and by comparing the map, the standardized map, with our results present on the gel electrophoresis, we can now detect the presence of the virus, if it is present or not. This figure shows where we prepare the specimen. We put the reverse transcriptase enzyme to transfer the RNA of the virus to DNA, and by special techniques and special preparation, it is put on this amplifier, which amplifies the number of genome of the DNA present in the specimen. And after it is complete, we stain by gel and run it through gel electrophoresis and detect the presence of the viral RNA. Finally, serology is a method where we do not detect the virus antigen, presence of the virus antigen or the nucleic acid, but we look for antibodies in the serum or in the CSF. Once more, I'd like to repeat that the virus never reaches the bloodstream, but when it stimulates antibodies, neutralizing antibodies are present in the blood on the serum of the patient. Antibodies to rabies in the serum or the CSF suggest rabies virus infection. This could be detected by indirect immunofluorescent technique. The indirect immunofluorescent technique is a sensitive, rapid test for detection of rabies IgM and IgG. Of course, you know IgM appears first in recent infection, and this is followed by IgG, which appears later on after convalescence or owing to vaccination schedule. Test specimens are reacted with rabies antigen on the slide, followed by addition of secondary fluorescent label anti-species antibodies, a specific immunoglobulin sites read by fluorescent microscope. This figure shows the indirect immunofluorescent technique. Here, the specimen is prepared on a slide. The specimen here is the serum or the CSF obtained from the patient. Serum or CSF is overlaid on a slide, fixed and prepared by special technique, and then we add specific antibodies, specific rabies antibodies. And then we add an anti-immunoglobulin, an interaction. If the antigen is present, an antigen-antibody complex reaction occurs and is fixed on the slide. This is followed by the addition of labeled anti-immunoglobulin labeled by fluorescent dye. If a specific antigen-antibody is present on the slide, these labeled immunoglobulins will interact, and you can see this reaction here present, demonstrating the antigen present on the slide reacted with rabies specific immunoglobulins, an anti-immunoglobulin labeled by fluorescent dye. And then after the preparation is full and furnished, the washing techniques and special techniques are applied. You examine by the ultra microscope for the presence of rabies antibodies. Next is the virus neutralization test. The virus could be, presence of antibodies could be achieved by serum neutralization assay performed in cell cultures to determine the presence of virus neutralizing antibodies. Thank you.